is it okay? Can I? Okay, so I'll try to just fill in the experimental details of what Julia said, because she explained all my results pretty nicely. But before I actually do talk about that, since this is a workshop, I wanted to just briefly mention something that really keeps us up at night these days. And it has to do nothing with the non-equilibrium stuff. And so before, so I guess I have to thank the people. So I've been spending the summer with Andreas in Munich, and it's been very productive. So I'll talk about those results largely. This work has been initiated by Tim Sanchez, and Steve is continuing it. And we are working very productively with Felix and Etienne, who are in the group of Andreas Bausch. But so what I want to say, just maybe take 5 minutes, is the other 50% of my lab, and what we've observed there over the past a year, and what we really don't understand. So at no point in my life have I seen so much and understood so little. So if you have any ideas, please let me know. So we start with just rods, and these are virus rods that are 1 micron long. And we add a depletion interaction, which is just a very generic interaction. And if you add enough rods, at certain point, you will observe a self-assembly of isolated 1 rod length thick smectic layer. So this is a smectic layer. This is maybe 20 microns in diameter. The rods point into the plane of the screen. And so the rod is 7 nanometers in diameter. And there's millions of rods here, okay? And so this is a single smectic sheet. And, we call it a colloidal membrane because in a certain way it's similar to membrane. And we do understand to a certain extent why this forms. But then we did a very simple experiment where we took 2 different rod lengths, a short rod and a long rod. And these are different by 30% in size. And we only labeled the long rods. And so I don't know if you can see that, but I'll switch the light on for a second. You have a membrane, and you have a dilute long rods in the background, and then they form these domains that are about 1 micron in size, and they contain about 10,000 viruses. And if you can wait for many, many days, and they will remain of the same size. And their polydispersity is 1%. So you essentially have assembly of 10,000 plus minus 100 rods. We do understand this to a certain extent, and it's a very nice model system. What you can do is you can visualize the formations of these clusters. Well, they are pre-critical and post-critical. You can measure their free energy, and you can show that as you increase the cluster size, the free energy increases. So this is the equilibrium size. And you can also, this is like a colloidal bead, so you can measure the interaction potentials. If you don't believe me,
I'm sorry, is this a cluster of long rods or short rods?
This is a cluster of short rods in a background suspension of long rods.
And are all the short rods in there? Or just
No, you can see this is a membrane. So you can see this dark, faint red thing in the background. And so there's about 80-20% difference, right, in concentration. So the concentration in a cluster is about 5 times higher than the concentration in the background.
What's the length ratio of the rod?
30% length difference. And they're different they're also different chirality. And so we understand this a little bit, but so you can do a very nice experiment where you take short and long rods and you just mix it and you wait for a while, and you see that you instead of just bulk phase separating like oil and water, you get formation of these clusters, and these clusters separate and form a real crystal. In this case, you can think of these things a little bit like type 2 superconductor vortices.
sorry, this depends on the chirality.
Yes. So one of these things is left-handed and the other one is right-handed.
If you do 2 left-handed or 2 right-handed.
Then we get a complete zoo of structures that we completely don't understand. So for example, we change molecular parameters. You see a nice little cluster. You can make it grow. It's a much floppier cluster, so you might think the line tension is, much lower. You make it grow even larger, and then something interesting happens where this cluster undergoes a polymorphic transition into this kind of horseshoe shape. If I make it grow even larger, it's going to get absorbed, the horseshoe is going to get larger, and eventually it's going to close into a ring.
So this is the homochiral part.
Yeah, this is very weak chirality, right?
What are you doing to make it bigger? You're adding more?
Yeah, I'm just, with the laser tweezer, I'm combining things and fusing them. That's not shown in the movies. And so at the end, you get this kind of two-dimensional vesicle that's embedded in a background membrane of long rods. And, so this is something that we completely don't understand. And then there's just a lot of phenomena that we don't understand. Each one of these clusters by itself is going to be spherical. If I take two clusters, they're going to deform, and they're going to very strongly bind to one another. This is tens of kT. So each one of these in isolation is a cluster by itself, and they deform and bind and form this kind of dimer. You can get, you can get in strain within this thing. And so here you, for example, can specifically bind a little droplet, and this is going to be very specific to the size of the small droplet. So it's a specific kind of lock and key binding. You can, start assembling structures of incredible complexity here. We have vesicles embedded in one another under different conditions. You can take these droplets and you can click them together to make higher order structures. And so, for example, this is something that my students did. This is a polymer. And so these are monomers, right? And they just connect them together into this polymer. And surprisingly, this polymer really wants to have 120 degrees with respect to one another.
The lengths of these rods is still the same.
We are changing here the molecular parameters, right? And so the problem that we have in the lab is that, we have dozens of rods, and we can mix any two things. And every time we do that, we observe a new kind of crazy thing. So we can generate new structures on a, month length scale, and understanding a structure is a year time scale. And so that's a non-steady state solution, which is a little bit problematic. But so I want to talk, so the rest of my talk is going to be about real non-equilibrium systems. Well, my lab, I guess, might be considered that. And so, so this is just one example of kind of being able to choose length scales and assemble things on very well-defined length scales. And what I want to talk about is, can you kind of systematically make this kind of structures that would not only be spatially varying, but it would also be varying in time domain, okay? And so, by definition, that's not going to happen in equilibrium, so we need to feed energy into the system. And so these are kind of our inspirations from a biological world. This is a sea urchin sperm. It's really an oscillator that beats at a certain frequency. And this is a cytoplasmic streaming where, you have a Drosophila oocyte, and you get this spontaneous circulatory flow that just keeps going in one direction. And so, as a physical thing, if you can reduce this to a few components, it's an interesting behavior that could be studied from a materials perspective. And
What's actually moving in the lab?
lipid granules, that's food for Drosophila to, keep multiplying. And if this is millimeters in size, and if you wait for these things to diffuse, the cell dies, right? So you need to steer the components together. So the common theme is, you have thousands of microscopic motors that Julia talked about, and they give rise to this complex behavior, and that's something that we would like to understand better. When I was a grad student, I actually spent 1 year trying to work with live cells, and so after that I abandoned all such efforts for a considerable future. And so what we decided, which is maybe simpler, maybe more difficult, is to really reconstruct this kind of behavior from the bottom up. So start with individual components. One of the components is microtubules, which is just filaments that are microns long. And the other component is motor proteins that consume ATP and take 8 nanometer steps along a filament length. And studying how one of these things moves has been studied in great detail by single molecule community, and there's 12 parameter models of kinesin stepping, and they've measured all the parameters. So understanding how one of this motor moves is understood in some detail, but how do collections of motors give rise to non-equilibrium phenomena is something that's not very well understood. So our motivation is really the work by François Nedelec and Stan Leibler. And so what they did is they took one of these motors, and by itself the motor just walks along the microtubule, and it doesn't do too much as far as we can tell. But what they did is they bound one of these motors into a cluster. So there's on average 2, 3, or 4 motors. And so you have this motor walking along this direction, this motor walking along this direction, and you push the filaments against one another, and you get interfilament sliding. And if you do that, you get interesting self-organization phenomena where you get formation of asters, okay? And so all the motors move towards the end, and you get these microtubules that are aligned outwardly, and the motors are concentrated in the middle. And so we were really motivated by these experiments, and we just changed one parameter, and that completely changes the behavior of the system. Kind of what happens with this, after the asters are formed, it's a quasi-static solution, right? Because nothing, you form these asters, the motors are concentrated here, and nothing happens. The system has spontaneously separated. So the only other thing that we add is we add a depletant, or we induce a microtubule bundle formation. So you have 2 filaments, and now they're parallel to one another, and that greatly increases the efficiency of motor sliding, because here I can walk, and there doesn't have to be another microtubule by, and so most of the motors, or a fraction of the motors, are just are moving along the microtubules without inducing any sliding. Here, what we have in a bundle is if the motor walks, it will fall off after a micron or so, but when it falls off, there's a lot of binding sites, so it rebinds. So by making a bundle, you're essentially increasing the effective processivity of the motor. Now, if you have polar arrangement, both motors are going to walk towards this end, and nothing is going to happen. There's going to be no microtubule sliding. If the motor are of the opposite polarity, this motor is going to walk here, this motor is going to walk here, and you're going to get sliding of 2 microtubule bundles. Now the question is, you want to build bundles, but you also want these microtubules to allow to slide past one another. And so that's not a trivial thing to do, but it's actually pretty easy. If you use cross-linkers, that would be pretty difficult. But if you use a depletion agent, it's a pretty easy thing to do. And so what you do is you just add a macromolecular crowding agent like polyethylene glycol. And if you have 2 microtubules, the polyethylene glycol is excluded from this region between 2 filaments. So you have a higher osmotic pressure on the outside than on the inside, and you get spontaneous bundling of microtubules. You can directly observe these bundles that are passive, and that's shown over here. Okay, so you have one microtubule observed in dark field. You have a short segment of another microtubule, and this short segment is just diffusing along the microtubule back and forth. And so what that tells you is that the friction between these 2 filaments is very low, and it's essentially hydrodynamic. So these things can very easily slide past one another, which is not the case for all the systems we studied. For actin, it's much different. And so you can analyze this kind of data in great detail. And the other thing that you can do is you can directly measure what is the adhesion strength. Okay, so you have 2 filaments over here. One is shown over here. One is shown over here. And they're in this bundling solution, and we keep them apart with a trap. Okay, and so these filaments want to maximize their overlap, right? Because that minimizes this energy. So they're pulling these beads out of the trap. And so you can do those experiments, and you can directly, so if I release this bead, the microtubule will attract, and it's going to maximize the overlap length. And this is what you can do, observe here experimentally, is we have one bead here, one bead here. We pull them apart. We let one of the beads go, and the microtubules retract. And then we pull it back apart, and we let it go. And so this actually tells you what is the force and what is the sliding friction.
Sorry, so
Yes.
you assume that the drag is dominated by friction on the microtubule,
Yes.
but not friction on bead?
Yes. We can actually do it by just applying a flow, so we don't have the bead attached to a microtubule when we are doing a dynamic experiment, right? So what you could do also is you can have a bead here, you can apply a flow, this is going to slide it away, and then you know, and so you don't need this a second bead in a real experiment. Are you satisfied, or?
I'm just wondering, it just seems like to actually extract the force, you have to have some kind of hydrodynamic model for...
Yeah, okay, so we do that. I don't want to spend too much time on that.
Okay.
What we do is we actually, let two beads jiggle around. You find their spatial separation, and the spatial separation fluctuates as a function of time. And, you can get a histogram. And then if you do it with a bead, you can see that the average separation between two microtubules is actually shorter when the microtubules are present. So this is the distance that the microtubules are pushing the beads together. And so this is a real equilibrium measurement. And by measuring the lambda, by knowing the trap stiffness, you can measure the adhesion force. And so this adhesion force is going to depend on depletant concentrations. But for the experiments that we work at, the adhesion force is 20 kT per micron, which turns out that the force pulling this, retracting this microtubule is 0.1 piconewton. It's pretty weak compared to molecular motors, so we don't really need to worry about it too much. And so this is an ideal bundling agent. And so what happens is, again, no polar arrangement, no microtubule sliding, antipolar arrangement, you get microtubule sliding. And so here we have a quasi-two-dimensional suspension. You have one microtubule shown over, a microtubule bundle shown over here, a microtubule bundle shown over here. You have, we don't know what the polarity is, but by themselves, these are static structures, so we assume that they're not polarity sorted. But they're going to diffuse around, and once they encounter one another, they initially slide off of one another, and they rotate by 180 degrees, and then they rebind, and they stay rebound, and they don't do anything after that until they encounter another bundle. And so what you want to do is you just want to increase the concentration, the probability of this kind of recombination occurring, and so you just increase the microtubule concentration. And so this, what we really don't understand is that most of the time we see these things, 90% of the time we see them extend. If you do the same experiment with actin and myosin, you'll see them contract. So that's a fundamental difference that probably has to do with the flexibility of actin compared to microtubules. And so if you increase the concentration, you start with something like this, and you can see that the system becomes unstable and starts generating this large-scale flows that Julia has talked about. And it goes on like this. What you would really like to do is you would like to see how this initial instability develops. And so the experiment we are doing right now is we're actually taking a shear cell and aligning it. And so you start with a very well-aligned initial state, and then you can see this bend instability develop, and the flow starts in this direction, okay? And so that's the instability that I don't have to explain because Julia has done a very nice job of explaining that. But so we hope to, by looking at this initial instability, that we can actually measure some quantitative parameters of our system. This is a very robust system. So this is a millimeter length scale, and this is a large scale pattern. And what you would notice is this is a piece of dirt that you always get in real experiments. But in this case, it's useful because this piece of dirt is tens of microns. It's completely non-Brownian, but right now it's being advected and it's moved, hundreds of microns in a matter of tens of minutes. And so, you know, so it's active transport, something that's reminiscent of a live cell, okay? So what I've shown so far is, so what I'd like to say is that what we actually spend a lot of, there's about 15 components in the system, and we've spent a lot of time trying to make the system robust. And so what we're able to do now is we can actually take an active sample that's fully doing its thing. We can freeze it at minus 80 degrees, and we can ship it to our collaborator in Germany, and he can just unfreeze it and start doing experiments. So that's a pretty, you know, so that's a nice thing for reproducibility and things like that. And so we hope that in the next year, you know, if people are interested, we can start shipping them the small quantities of the samples. That's something that's feasible now, and it wasn't before because you would need to, you know, mix 15 components, and even I cannot do it. Only my grad students can do it. I tried to do it last year, and I failed. So the important properties are what are microscopic microtubule dynamics, and what are the macroscopic properties, okay? So if you really zoom in, you start seeing individual filaments or bundles of filaments, and this, I think, illustrates what's happening microscopically in the sample. You have these bundles, right? And as they merge, you can see that they extend, and as they extend, since now they're coupled to a network, they buckle. So here you have, you have an extension and you have a buckling event. And so once you buckle, you generate fragments that are free to associate with other filaments. So here you see merging. As it merges, it buckles and it fractures, and then it's free to reconnect with other pieces of filaments, okay? And so this is shown in this movie in this time sequence, where you just have filaments coming together, and as they come together, you can see this region is being elongated because it's getting dimmer, and then eventually it fractures and you generate new pieces. So the system wants to macroscopically polarity sort. If it ever would manage to polarity sort, it would just die, right? Because there would be no interfilament sliding. But it never gets there because there's this instability to where, you know, it buckles and breaks, okay? And so it's a very well-controlled system. And so, you know, by tuning the ATP concentration, you can follow the traces of beads. This is passive beads. They're not doing too much. And at saturating ATP concentration, the beads are essentially being advected along a straight line. And you can measure mean square displacements of these active gels. So 0 ATP concentration, it's a little bit subdiffusive. And by increasing ATP concentration, you get super diffusive. And at highest concentration, it's completely ballistic. This is slope 1, this is slope 2, and you saturate, of course, but we can't track the beads for such a long time.
Sorry, it's ballistic, presumably only after time scale it's related to the correlation.
Yes, yes. But we can't, you know, they go out of focus, and so to get long MSDs, we need to follow them for a long time.
Longer than the correlation.
Yes. Just
Shorter than the correlation.
yes. All right, whatever, you're correct, whatever you, one of these two things is correct. Okay, so what I've done so far is describe so I've described this really what are at first taught to us as appeared as isotropic three-dimensional gels. And the idea was, well, let's try to increase the microtubule concentration. And so we were not able to do that yet in 3D, but we can do it in 2D. And the way we do that in 2D is we have an oil surface, we have water on top of it, there's a surfactant that stabilizes this surface, and microtubules are being depleted onto this oil-water interface. And so at very low concentration, you just get individual microtubules that lie at the interface. So the oil is below, the water is above, and the microtubules just lie at the interface. If you increase the concentration, here what you get is longer filaments, right? But you can still see our bundles. You can still tell their beginning and end. And then at some point, the system starts percolating. And so you get this kind of percolating filaments that look very much like 3D filaments. And so I think this is a real kind of phase transition that you can characterize. But then if you increase the concentration even further, the bundles become thicker and thicker, right? And so the bundle thickness increases, and here you have what you would call a liquid crystal. But I don't know if, there doesn't seem to be, to me, a transition between something like this and something like this, right? The bundles just increase in thickness. And so I don't know how to, if this is really a phase transition or not. I don't think there is a real phase transition here. And the dynamics of this would be identical to the dynamics I described before. And what is the dynamics of this? And so this is something that people have, these are your disclination defects that people have studied in great detail in equilibrium liquid crystals. And so because it's a 1/2 defect, you know that this is a nematic order. So in principle, in active liquid crystals, you can have either polar order, where all the filaments point in the same direction, or you can have nematic order, where half of the filaments point upward and half of the filaments point downward. And so, if you have a polar vector, a vector that has, that has an arrow with it, you can't make a defect like this because you don't end up with the same state. And so the only way you can make, the only defects you can make are defects of charge 1. And so polar liquid crystals form +1/2 and -1/2 disclinations, while nematic liquid crystals form something, these 1/2 disclinations. And so the observation of these defects really shows us that our nematic liquid crystal is, that it's a nematic liquid crystal, and that makes sense because our fundamental building block is an extensile bundle. And so you get, this kind of behavior of the system, and in a certain way, this behavior is very similar to, the dilute system I've talked about, right? Is what's happening here is you get this dynamics, is that you get these +1/2 and -1/2 defects throughout the system, and the so what's known and what Sriram shown before is that the defects in active liquid crystals are spontaneously motile. So by themselves, and this is an experiment on granular rods, you have a +1/2 defect that's spontaneously moving in this direction. I don't know if this is in the right time dimension. I think I rotated by 180 degrees. So it's moving in this direction, while a -1/2 defect is just statically sitting here, and that's because this is a symmetric structure and this is an asymmetric structure. And so what's happened in our system is you get this, in these nematic liquid crystals, you get a band instability, right? And so once you get this band instability, it increases in amplitude, and as a function of time, you get fracture of a nematic director here, and as this defect is being propelled in this direction, this fracture line heals, and you get an unbinding of -1/2 and +1/2 defects. And so the steady state dynamics is determined by, the spontaneous generation and annihilation of these defects. But it's a little bit of a complicated system to work with. It's very chaotic, and so if you think about equilibrium liquid crystals, is what's really important is your ability to create monodomain samples and really introduce defects in kind of a controlled way. And so we are not able to do that in 3 dimensions, in these flat 2-dimensional disks. And so what we are focused on is really trying to make better controlled geometries in which we can really control the number of defects and study them in isolation. And so from equilibrium studies, it's very well known that if you take a crystal or a liquid crystal, and if you try to cover a sphere, you cannot do that. And so you need to introduce defects that can be either point defects or scars. And if you take a liquid crystal, you need to either introduce these kind of +1/2 defects on both poles, or these can split into 1/2 disclinations. That was work done by David Nelson. And so most of these problems are, static defects on non-deformable surfaces. Okay, and so you have a recipe of how to solve an equilibrium structure like this. The topology demands a certain number of defects, and then you minimize the free energy, and that determines the exact location of the defects. Now, if you have an active defect, you have elastic on a spherical surface, for example. You have elastic interactions, but in addition, these defects move with a constant speed. And so you have a problem is, can I put defects on a tetrahedra, for example, and have them move at a constant speed? Moreover, these defects move in preferred directions, right? So the question is, these are the three things that you ideally want to satisfy, and what would be a dynamical trajectory of defects on a spherical surface that's deformable? And so if you want to cover something like this with a defect, if you want to cover a disk with a defect, you have to introduce plus 1/2 defect here, and you get into that's just something that topology demands on your sample. And if you have a sphere, you can either have these 2 plus 1/2 defects on both poles, but in most cases this is actually an unstable solution, and you get these 4 1/2 disclinations on these corners of an American baseball. This was done in U.S. By David Nelson. Oh, okay, yeah, okay. I mean, okay, so you can. So you can put it on a spherical dome, and you can see that you actually get spontaneous circulation of active liquid crystals in one direction. And so that's very different from what happens in bulk, right? And this circular flow is going to persist for a very long time. And so you can, if you at any point stop it and count the defects, you'll actually see that here you have 3 plus 1/2 and 1 minus 1/2, as is shown over here. Yes.
So if you had a very big system,
Yeah. Yes.
there'd be some characteristic distance between
Yes. Yes. Yes.
between disclinations.
Yes. You would start generating more defects. Here I'm generating 2 additional defects. I think if we can make it smaller, we would never generate an additional
No,
defect.
but I want to ask about going the
Yeah.
other way.
Yes.
Suppose you had a sample. I'm thinking that this sample is not much bigger than typical spacing between disclinations
Yes.
in a very big sample.
Yes. Yes.
So as a result, you're actually getting coherent motion across the whole
Yes.
sample.
Yes.
But suppose you made the thing very big
Yes.
compared to the characteristic space
Yes.
between disclinations. Will you only get motion along the surface and the bulk be disordered?
I think so. Yes.
Okay.
Uh,
That's what I would have thought.
In that case, the field of view is more or less the size of the droplet.
This is not a droplet. This is a 2-dimensional dome surface. So this is.
Yeah, that's what I meant.
Yeah.
The field of view, like compared
Yeah. Yes. Yes.
You're seeing the whole drop.
Yes. Okay, so now we go into deformable 3-dimensional vesicles. So if a vesicle is very small, you actually observe 2 plus 1/2 defects at both edges. And this kind of vesicle exhibits interesting dynamics. So what's happening is that it's being extended, and these things are pushed apart, but then it reaches a critical buckling wavelength, and then it folds on itself, right? So you have this 1/2 defect, 2 plus 1 defects, and both ends, and they just are pushed apart, and once you reach a critical length scale, it folds on itself, and this continues on and on, this kind of dynamics. If you increase the vesicle size a little bit, you get what we call ring modes. Is essentially you get just on an equator, you get a ring of microtubules, and this can undergo a bending instability where it escapes in a 3rd dimension, and but then it comes back into this ring mode, and you get circulatory flows in this direction. We haven't studied these 2 things in such great detail, but then if you go to the large vesicle, what you get is you get 4 plus 1/2 defects. And if I could look from the other side, I would show you that there is, a 4th defect as demanded by topology, because topology doesn't care about, whether something is equilibrium or not.
But again, assuming you make this bigger, you get.
Yes, yes. And so what you can do is you can make a bunch of these vesicles, and you can count their probability, and you can see that for small radii, you get only these kind of lemon-shaped vesicles, At intermediate sizes, you get a mixture of these lemons and rings and these 4 + 1/2 defects. And if you go above a certain size, you only get 4 + 1/2 defects. And that's something that that's something that we are studying right now in the lab, is what is the dynamics of these 4 + 1/2 defects on a sphere. And so this is an example of a vesicle, and these are 6 different projections. So this is from the top, this is from the bottom, and this is from the 4 sides. Okay, and so you can directly track the motion of each defect, and if you see it from more than 2 directions, you can determine its position. And so that's something that my student did. And so this is a spherical vesicle, and so you have 4 defects, and they're just moving around on the surface. And the question is, what is the trajectory of these defects? Is it completely chaotic, or is it something regular? And this is the data that Felix from Andreas's lab just sent me this week. And so what we seem to see is 2 kind of configuration and a very periodic switching between 2 configurations. So one configuration that's very prominent is planar configuration. And so what he plots here is 6 angles between any 2 points. So in a tetrahedra, you can have 6 angles between any 2 points, and the 6 angles would all be equivalent to one another. In a planar configuration, you would have 4 90-degree angles, and then an angle between here and here, and here and here would be 180-degree angle. And so this is a time trajectory of all these angles. And so you can see that so this is when it's in a tetrahedral configuration, because you have 2 peaks, and then it switches to planar configuration, and then it switches back into tetrahedral configuration, where you again see 2 peaks that are at maxima, and all the other ones are much smaller. And so this is the periodicity, and this periodicity is very regular. And so you get this switching between 2 different states, and if you count the histograms, you see the red histograms correspond to these planar configurations, and the blue histograms correspond to tetrahedral oscillations. And that's what you can nicely see on this chymograph. So these are the 6 angles, and then you can see that at any point, 2 angles are approximately 180 degrees, and you see these very periodic patterns, but it doesn't seem that there is any, it can choose any 2 points to go into this 3rd dimension. And so finally, what I want to say is, if you take one of these vesicles and then you can make it floppy, you can control the surface tension. And so we start with our spherical vesicle, and we change the osmotic pressure, so we are taking the water out, and it becomes floppier. And at some point, it starts doing funny things, okay? So you get 4 of these defects, and they deform the vesicle, and they start moving around as is shown over here. And this is quite robust, and then if you change the osmotic pressure back, you would get a perfectly spherical configuration. But this does look like some kind of animal that moves around, but it's just it's 3 components. And so that brings me to the end of my talk. And so, I mean, so I think, from a practical perspective, we have a very kind of robust system of extensile bundles, and we really works every time in our hands now, and that's something that took a lot of effort. And you get very complex behavior by really starting with very, very simple components, and that kind of mimics some biology. And so this is another example that you can get oscillations, is you get these bundles that are lodged at the interface, and they start oscillating like a ciliary beating pattern. And so my student actually won an iPad for this, so he was very happy. Thank you.
Time for a couple of questions.
Hi, very beautiful. I guess I have a question about the comparison with old experiments by Nedlek. I mean, it's not clear to me why just putting more putting more interactions between the things would change something from being a polar liquid crystal to be a pneumatic liquid
So,
crystal?
so
Because
what I
the
have,
symmetry is
yes.
pretty much the same.
So,
in
so
that point.
what I haven't said is that microtubules are an extremely painful experimental system, and so experimentally, what you find out is that they stick to one another, okay? And so Nedelec used very long filaments that are somewhat, I would say, sticky. And in our case, we actually have to make our individual microtubules are 1 micron in length. The characteristic size of this bundle is 50 microns, right? And so it's a very soft bundle. And so what I think we are changing is we are changing actually cross-linking density between filaments, okay? And if you have 2 1 micron filaments, you know, maybe a couple of them will be cross-linked, but that's not going to really matter, okay? And we can see if we start introducing cross-links, you get kind of contractile behavior.
Hi. I was wondering, in the planar experiments,
Yes.
Did you measure either the total amount of nematic order as a function of scale or the nematic orientational correlation function as a function of distance? Is it ultimately in an isotropic phase or is it in a weakly nematic phase, power law phase?
I mean,
something?
we're going to do it.
Maybe I asked you this before. I don't
Yes,
remember.
you did
Oh, I
2
already
weeks
did.
ago.
Sorry.
My student is not,
Forgets.
he's good, but he's not that fast.
Okay. I thought it's worth.
I will do it by September 1st, okay?
Could you comment on the dependence of characteristic lengths on concentration of microtubules?
Well, I mean, so that's that experiment that I've shown over here, right? Is, you know, in 2D. Huh? In 2D, we can systematically change. Okay, this experiment over here, right? So here we can systematically change microtubule density, right? And so it seems that, you know, maybe there is some difference in density, but it's, you know, I believe that these are all the same length scale. So we should actually do that systematically and see how it changes with microtubule concentration.
The length scale is increasing with the concentration, right?
Yes, it seems so, but we haven't quantitatively measured that, but we should do that.
Time for one more question.
Are your vesicles fluid or elastic?
They are fluid. I mean, in plane is fluid.
In plane, in shell? In shell?
Yes, yes.
I just have a really quick question. Did you tell us how you encapsulated the microtubules in your vesicles?
No, I didn't,
Can you?
but I can tell you. So this is the method that was developed by Gladys Messiera in France. And so it's essentially a double, you know, that's what Andreas is an expert on, or his student is. You essentially have a oil droplet that's coated and with the lipid, and then you go through an interface, and they have a robust way of making it.
Put a solution of tubulin in.
Yes, yes, yes. Within an emulsion, and then you pass it through oil water.
After you.
Yes, yes. It's very robust. It works really well. I can give you a reference if you want.
Okay, let's thank both speakers. Really.